We ended the last lecture by looking at how synapses work. Now, remember that what a synapse is a specialized connection between one neuron and another, essentially the way that neurons talk to each other. Our discussion of synapses last time completed our review of those three major functional specializations of neurons that I outlined 2 lectures ago. These specializations included, you'll recall, first, the ability to produce an action potential. That's the rapid and stereotypic change in membrane potential that neurons use to send signals. Second, the ability to propagate that action potential from one place to another along the long axon of a neuron. And third, the ability to cause a change in the membrane voltage of another cell. And that's what the synapse was responsible for doing. But even with these remarkable properties of neurons, we're still a very long way from having the basic elements that we need for a functional nervous system. We now understand something about how neurons can convey information from one part of an organism to another, but there remain 2 other critical features of how nervous systems work that we still need to consider. Not only do we need to be able to transmit physiological information, we also need to be able to process that information and to store it. And these are the 2 issues we're going to focus on in today's lecture. First, we'll ask how neural information is processed and integrated. Of course, the way that information is processed by very complex nervous systems, such as our own, or even the nervous systems of very simple animals, is beyond the scope of an introductory biology course, but we can illustrate the most fundamental principle underlying the processing of neural information, which is based on a process called synaptic integration. And that'll be our first topic. Second, we'll look at an example of the kind of mechanism that's thought to be involved in the storage of information in nervous systems. Again, a comprehensive review of the mechanisms of learning and memory is beyond the scope of this course, but we can explore one current model for the cellular basis of memory, which will serve as an illustration of the kind of mechanism that must be necessary to provide a physiological foundation for the storage of information by nervous systems. Now, to begin our look at the mechanisms of information processing in nervous systems, let's go back and quickly remind ourselves about what we know about synapses. I pointed out last time that most synapses are chemical synapses. Remember that in a chemical synapse, an electrical signal, that's the action potential traveling down an axon, gets converted into a chemical signal. The chemical signal is the release of neurotransmitter, a chemical that's released by the presynaptic side of a neuron in a synapse. And that chemical signal then goes to the postsynaptic cell, the next neuron in line, and induces an electrical signal. Now, the way that the neurotransmitter, the chemical signal, does this is by opening ion channels on the postsynaptic cell membrane. And the way that it does this is by basically using the same kind of cell signaling mechanisms that we looked at a number of lectures ago. The neurotransmitter binds to receptors on the postsynaptic cell's membrane, and those receptors, through a variety of mechanisms, will open or close gated ion channels. Now remember, those ion channels change the permeability of the cell membrane, in this case the postsynaptic cell membrane, to various ions. And if, for example, an ion channel opens that's permeable to sodium, a positive ion, it will rush into the cell, changing the membrane potential in the postsynaptic cell, depolarizing it. And thus, the chemical signal gets translated into an electrical signal on the other side of the synapse. Now, there are 4 other points that I want to make about how synapses work that will be useful for our discussion of synaptic processing. The first is to simply remind you that synapses are very small, very specialized, and highly localized structures. Synapses usually occur on, remember, what we call the dendrites, or the cell body of a neuron. So an axon will send its connection and make a connection, a synaptic connection, on another neuron on those shorter processes that extend out from the cell body, called the dendrites, or right on the cell body itself, but not on the axon of another neuron. The second point is that the change in membrane potential caused by the firing of a synapse happens very quickly and also only lasts a brief period of time. Now, it can happen very quickly because the chemical signal the neurotransmitter that's released at the synapse has to travel a very, very, very small distance. In fact, the width between the two cells, the distance between the two cells, the presynaptic cell membrane and the postsynaptic cell membrane at a typical synapse is only about 20 to 40 nanometers. That's 20 to 40 billionths of a meter. That's a very, very short distance. And so the neurotransmitter diffuses across very quickly, and a synapse can work within a millisecond or so. Now, more importantly, the effects of the synapse firing are very brief. That depolarization only lasts a very short time, and this is because there are a number of mechanisms that will either take the neurotransmitter back into the presynaptic cell or break the neurotransmitter down, so that the depolarization that the neurotransmitter is causing will only last for maybe just a couple of milliseconds. Now, the third point I need to make is that different kinds of synapses can be either excitatory or inhibitory. Let me explain that a little bit more. What that means is that if a synapse is excitatory, then it excites the postsynaptic cell, which in electrophysiological terms means that it makes it more positive. Remember that the cell membrane is usually negative inside relative to out. So if it makes it more positive, or what we called last time, or referred to last time as depolarizing the postsynaptic cell membrane, then it brings it closer to the threshold value that's necessary to be reached for that second neuron to fire its own action potential. That's an excitatory synapse. Now, an inhibitory synapse does just the opposite. An inhibitory synapse will make the postsynaptic cell's membrane even more negative than the resting membrane potential. In other words, it takes it farther away from what would be the action potential's threshold level, and thus makes the second cell, the postsynaptic cell in the synapse, less likely to fire an action potential. Now, how excitatory and inhibitory synapses work really is a matter of the nature of the neurotransmitter and of the receptor that neurotransmitter binds to, and most importantly, of the ion channels that are opened and closed by that neurotransmitter binding. So, for example, if a neurotransmitter has the effect of opening channels that are permeable to sodium, well, then sodium rushes into the cell, and you'll recall that that causes the cell to depolarize, and that would be an excitatory neurotransmitter. But it's equally possible to have a neurotransmitter that causes channels that are normally open and that are selective for sodium to close in response to the neurotransmitter, and that would decrease the permeability of sodium, and you'll recall from last time, that would make the cell's membrane even more negative. So that would be an example of an inhibitory synapse. Now, we don't have to know the details of how excitatory and inhibitory synapses work. What we do have to keep in mind, however, is that a synapse can either bring the postsynaptic cell closer to its threshold for firing an action potential, or drive it to become further away from its threshold. Now, there's one final point I want to make about how synapses work, and that is to point out that the depolarizing or what we would say hyperpolarizing effect. Hyperpolarizing is driving the cell's membrane more negative. The depolarizing or hyperpolarizing effect of a single synapse firing is relatively small. In fact, it's very small. Recall that I told you that the threshold potential for a neuron to fire its action potential, in other words, the amount of depolarization that has to occur, is often on the order of maybe 15 or 20 millivolts more positive than its resting membrane potential. In other words, the potential across the membrane has to increase, has to become more positive by about 15 or 20 millivolts. A typical chemical synapse will only change the postsynaptic cell's membrane potential by just a few millivolts, 1 or 2 or 3, maybe 4 or 5, but not enough to actually cause that cell membrane, the postsynaptic cell membrane, to reach its threshold. And this is a critical point because it means that any one synaptic connection has a relatively negligible effect on the behavior of a postsynaptic cell. So then that raises a question. If a presynaptic neuron can only have a very small effect on a postsynaptic neuron, how does an action potential ever get generated in that second neuron? Actually, the answer to this question is the key to understanding the fundamental mechanism of information processing in nervous systems. In short, the answer has to do with the fact that any given neuron will typically have not just one, but maybe thousands of synapses made on it. Whether or not a particular neuron fires, then, depends on the summed activity of all of the different neurons that are connected to it. And this is the process that we call synaptic integration. Well, let's look at synaptic integration in just a little bit more detail. Individual action potentials, which are really something you can think of as the basic currency of a nervous system, are all or none events. That is, that they're like the ones and zeros in the digital code that a computer could use. A neuron will either fire an action potential or it won't fire an action potential. This is like a bit of digital information in computer coding. You'll either have a 1 or a 0. But whether or not a particular neuron actually does fire an action potential depends on not just a single input, but a complex set of inputs, allowing for an exquisite amount of processing and integration of information. In other words, information can come from several sources, and the postsynaptic cell can essentially make a physiological decision, if you will, as to whether to fire or not, depending on the information it's getting from all of these sources. As I said, there could be dozens, hundreds, or even thousands of synapses made on a single neuron. These synapses generally come from different input neurons. And often these different input neurons are sending information from very different parts of the nervous system or other parts of the organism. Thus, at any one particular time, the membrane voltage of a single, what we might want to call an output neuron in some synaptic integration circuit, is being affected by many different inputs, some of which are excitatory, some of which are inhibitory, some of which fire at the same time, some of which fire at different times. So the question then is, how are all these different inputs integrated to determine whether or not that output cell will fire its own action potential? Well, the basic mechanism by which synaptic integration occurs is called summation. To understand how summation works, let's start by just considering a fairly simple case, and let's consider just a case that has excitatory inputs alone. Now, recall the effect of any single excitatory synapse on a postsynaptic cell is almost invariably subthreshold. Subthreshold just simply means it's not enough to bring that postsynaptic cell to its threshold for firing an action potential. But the cumulative effects of several synapses can summate to achieve a depolarizing effect that does bring the postsynaptic cell to its threshold for an action potential. That's all that summation means. Now, summation can occur both in time and in space for a particular postsynaptic cell. Let's talk about what we would call temporal summation first. Temporal summation refers to the case of a single input synapse firing more than once and in sufficiently rapid succession so that the depolarizing effects of the first synaptic event haven't worn off yet completely before the second synaptic event occurs. In other words, the second depolarization can build on the first, bringing the membrane, the postsynaptic cell's membrane, closer to its threshold for firing an action potential. Let's think about this for a second. Imagine we're recording from a neuron, and we're doing this by putting electrodes, as we did a couple of lectures ago, on the inside and outside of the cell so we can measure the voltage difference across the membrane. Now, if we're recording in this way and we have a single synapse fire, we would measure, and it's an excitatory synapse, we would measure some degree of depolarization. In other words, if we were looking at this on an oscilloscope, for example, we would be watching the trace and it would become a little bit more positive just for a couple of milliseconds, and then it would go back to its resting membrane potential. Now let's imagine that we have one synaptic input into this cell, and we cause that synaptic input to fire repeated times. We can do this, for example, by causing that presynaptic cell to fire with a set of electrodes we've got at its cell body, where we're adding current, as we've done before, and causing it to depolarize and fire an action potential. So we're now going to have a series of depolarizing events occurring at that one particular synapse. Now, if we just have this series of depolarizing events occurring at some, let's say, relatively slow rate, what we would see is that each time the synapse fired, there would be some depolarization, and then that depolarization would gradually, in a millisecond or two, go back down to rest. And then there would be another depolarization, and essentially we would see a bunch of bumps on our oscilloscope trace, or a series of positive going deflections. But now imagine that we cause that presynaptic cell, cause that synapse to fire more rapidly. And specifically that we caused it to fire sufficiently rapidly that the depolarization of the first synaptic event hadn't yet gone back down to rest, but was still a little bit more positive than rest. What would happen is that the second depolarization builds on that. So, for example, if the first depolarization brought us to a peak of maybe 3 or 5 millivolts more positive than rest, if we fired the synapse again soon enough, then the second depolarization might build on a depolarization that's already 3 or 4 millivolts above where rest is already. And it would get to a higher peak, something maybe around 8 or 9 millivolts more positive than the rest. Now, of course, the details of the numbers don't matter. The point is that because it takes a finite amount of time for the depolarization of one synaptic event to wear off, we can build on that depolarization by firing the same synapse more rapidly. So what does this buy us? This means that we have a simple start to a processing system. Even if we only have one synaptic input to a single postsynaptic output cell, the rate at which that synaptic input fires could determine whether the second cell itself fires an action potential. If the rate is slow, then the second neuron won't fire. And if the rate is fast enough, then the second neuron will fire. So just the rate of an input can cause a decision, if you will, on the part of the second cell to fire or not through temporal summation. Now, we can also sum synaptic inputs in space. To explain this, imagine now that we have our same neural setup with our electrode, but instead of just one synapse impinging on that cell, we now have 2 different synapses. 2 synapses coming from 2 different cells. As before, if either of those synapses fire alone, the depolarization is unlikely to be sufficient to cause the postsynaptic cell itself to depolarize enough to fire an action potential. But imagine if those 2 synapses fired at the same time, or relatively close in time. Then their combined depolarization would sum again. And this summation, the 2 inputs occurring at once, could be enough for the second cell to come to a point where it reaches threshold and fire its own action potential. What have we gotten here? Well, now we have the ability to integrate information from 2 different sources. Imagine in the simplest of neural systems that you have an output cell which will fire only if 2 inputs fire at the same time. It's not much processing, but it's a beginning, and it represents the basic way that neural processing occurs through synaptic integration. Well, finally, let's consider a somewhat more complicated case. Now let's throw an inhibitory synapse into our circuit. Now imagine that we've got an output cell that's got 1 or maybe several excitatory synapses on it, but also has an inhibitory synapse. Now, in this case, the excitatory synapses, either through temporal or spatial summation, might have some kind of activity, some range of activities that will trigger that second cell to fire an action potential. But if the inhibitory synapse fires as well at the right time, it could cancel the whole thing out. It's another layer of complexity that we can add to figuring out how we can integrate inputs. In this case, we can have an input from some source that negates the whole deal. Regardless of how, or in spite of sufficient summation from excitatory inputs, a negative input, an inhibitory input, could cancel that. Again, an example of a relatively simple mechanism for integrating input from multiple sources in a way that essentially makes a single cell an integrator that allows for complex processing, in fact, providing the fundamental mechanism for information processing that is at the basis of all neural function, really. Whether we're thinking about a relatively simple animal such as a jellyfish, which has a network of loosely associated neurons that control the way that it undulates, or something really, really complicated like the 100 billion or so neurons that make up a human brain, the way that these neurons essentially make decisions has to do with integrating inputs from different sources. Of course, in the case of the human brain, the sources can be an enormous number of sources, and the kinds of integration that occur is something that would be beyond really still our ability to computationally determine whether a single synapse will fire or not. But the same mechanism applies. All right, so now we've seen sort of the simplest way of thinking about how nervous systems can process information, more than just transmit it. Let's turn now to that other feature of nervous systems we want to touch on today, which is how nervous systems can store information. Remember, it's the storage of information that provides the foundation for what we would call learning or memory. And of course, in nervous systems, this is a very, very important adaptation. Obviously, in our own experience, our own personal experience, learning and memory are a quite complex phenomenon. And we actually know quite a bit in the human brain about how these abilities are supported by equally complex anatomical and physiological specializations. Well, that's something that we don't have time to get into here, but just as we did to understand how nervous systems process information, we can consider a very simple case to illustrate the basic mechanism by which a nervous system might store information. And actually what we'll see here is that we can return to understanding something about the function of gated ion channels to gain insight into the basic cellular mechanism for information storage in nervous systems. Now, the history of thinking about mechanisms of learning and memory really date back for thousands of years. And in fact, modern thinking about it dates back for hundreds of years. But we're going to start our story really in modern times with a proposal made by a neuropsychologist named Donald Hebb in 1949. Hebb was a psychologist. He was trained as a psychologist, but in the 1940s there were a growing number of psychologists who became convinced that psychological processes were best understood in physiological terms. This was the time when the basic mechanisms of how nerve cells worked, how synapses worked, were all being figured out, and the neuropsychologists were convinced that we had to turn to physiology to understand psychological principles. Now, Hebb was particularly interested in memory. And he proposed that memory could be modeled in terms of changes in the way that assemblages of cells interacted with each other, and specifically in terms of changes in the relative strengths of synapses made between two cells. Now, the idea here is that some experience, which really on a physiological level would be some activity of that synapse, somehow would have a permanent effect on the strength of the synapse. We refer to this model now as a Hebbian synapse, and in fact this idea really has dominated thinking about how the cellular basis of memory can work ever since Hebb first proposed it. So how could a Hebbian synapse work? How can a synapse change its strength as a result of prior activity? Well, there are a number of mechanisms that have been proposed, but today I just want to talk about one that's actually becoming fairly well understood and fairly well accepted. And to understand this mechanism, I have to give you just a little bit more background about synapses. And specifically, I want to tell you about one particular kind of neurotransmitter found in vertebrate animals called glutamate. Glutamate is an excitatory neurotransmitter. In fact, it's the principal excitatory neurotransmitter in the central nervous system, that is the brain of vertebrate animals such as ourselves. As is true for many kinds of neurotransmitters, there's actually more than one kind of receptor that will respond to that neurotransmitter. This is true in general, and for many neurotransmitters, a single neurotransmitter can activate different kinds of receptors, and most importantly, these different kinds of receptors will then in turn open or close different kinds of ion channels. Now, in the case of glutamate, these two receptor types that I want to tell you about are called the NMDA glutamate receptor and the AMPA or AMPA glutamate receptor. Now, those names come from, the origin of how these receptors were discovered as being different, which is based on different pharmacological agents used to identify them. And we don't need to know about those details. What matters to us is that these two receptor types work in slightly different ways when they bind the neurotransmitter glutamate. Both NMDA and AMPA receptors open ion channels that allow sodium to cross the postsynaptic cell membrane, and so that's why they're excitatory. But NMDA receptors differ from AMPA receptors in two critical ways. First, the channels that are opened by NMDA receptors also allow the ion calcium to enter. So they're not just specific to sodium, they also allow calcium to pass. Second, NMDA receptors are actually inhibited or blocked by yet another ion, the ion magnesium. Now, there's almost always some magnesium present, and what that means is that in general, NMDA receptors are almost always blocked. But what unblocks the NMDA receptor, basically by driving off the magnesium, is depolarization. Now, this is kind of interesting because these receptors are going to open channels that will depolarize the postsynaptic cell membrane, but they can't do it unless the postsynaptic cell membrane is already partially depolarized. This set of facts, the fact that NMDA receptors allow calcium to pass and that they'll only open if the cell membrane is already a little bit, at least, depolarized, provide a key to understanding how NMDA receptors might provide a molecular basis for something like a Hebbian synapse. And here's the way it's thought to work. A neuron having NMDA receptors may only have those receptors open if that same neuron gets coincident activity, simultaneous positive or excitatory input from some other source. If it does, then the NMDA receptors will open. That will further depolarize the cell, but more importantly, it allows calcium to rush in. And the model for how NMDA receptors might then work to affect long-term changes in a synapse is through the action of calcium as a second messenger. Remember, we've talked about calcium as an important second messenger in intracellular signaling, and it is a very ubiquitous second messenger. Calcium, when it rushes in, triggers a whole series of intracellular responses that will change, essentially, the synaptic machinery of that particular cell. So if the NMDA receptor opens, allowing calcium to flow in, then that cell will strengthen its synaptic machinery through the action of calcium as a second messenger. That's the model. Are there synapses that actually work like this? Well, actually, a number have been discovered, especially in vertebrate brains. Synapses that show what we call a phenomenon, or the phenomenon called long-term potentiation. Now, most neurons, if you depolarize them repeatedly, if you put an electrode in a typical neuron and you just keep causing it to depolarize, eventually the depolarization will run down. The neuron essentially gets tired. I mean, it physiologically habituates. On the other hand, synapses involved or that exhibit long-term potentiation show just the opposite. In these synapses, if the synapse repeatedly fires, the response of the postsynaptic cell actually gets stronger and stronger and stronger. Well, it turns out that synapses that show long-term potentiation have been shown to have, for glutamate, both AMPA and NMDA receptors. And the model is this. As the synapse fires, that will open the AMPA receptors. Remember, those are just responding to glutamate, allowing sodium to rush into the cell, depolarizing it, sort of like any good old excitatory synapse would. But if the AMPA receptors are stimulated enough, for example, by repeated or rapid firing, then the level of depolarization of the postsynaptic cell gets to be high enough that the magnesium is driven off the NMDA receptors. The NMDA receptors now can respond to that same glutamate signal, allowing calcium to rush in, and as calcium works as a second messenger, essentially remodeling that synapse. And what you in fact find is when that happens, these long-term potentiation synapses not only increase in their strength, but actually maintain that increased strength for an extended period of time. Just as Donald Hebb had predicted, the synapse's activity, which would correspond to some experience, changes the strength of that synapse over the long term. Again, it's not much of a step towards understanding learning and memory, but it's an important first step because we can take this basic circuit of a nervous system, a single synapse, and cause a permanent change in the way that synapse works from a low activity state to a high activity state through the action of NMDA receptors.